trained as a biochemist in Canada. I came to the States and learned molecular biology. I've been involved in the Human Genome Project with a couple of Nobel laureates, and a lot of the development of essentially the tools we use for microbial ecology and for DNA sequencing. I actually worked at NIH for a couple of years. I set up the center at NIH, the Human Genome Center, when it first established there. I worked with a Nobel laureate at Harvard. We set up and sequenced a small bacteria at Harvard. And eventually, I found myself here at George Mason. That basically, from any of these buildings, right, have designed a couple of them. And what was being established at that point was the biomatrix, and really it's what you call now systems biology. So although I've got a lot of background in technology, half of what we do, or 3/4 of what we do now is actually mathematics, modeling, ecological systems. So we'll talk a little bit. I'm going to try to keep this at high level. I'm not sure if this should have been after dinner or before dinner. We'll go from there. Okay? I'm going to give you a couple of concepts on what we mean by the Human Microbiome Project. Talk a little tiny bit of how we do next-gen sequencing. A little bit about the concept of molecular systematics. That is the framework that we use to basically identify the bacteria and try to look and see how they're functioning. The concept of systems biology and the modeling of nonlinear systems. I'm going to just give you a little blurbs from a project on how the microbiome is involved in obesity, non-alcoholic fatty liver disease, which is a big silent epidemic in this country, so is obesity. Talk a little bit about how the microbiome interacts with the brain. This is a big new field now, the gut-brain axis. And then I'll talk about, this is kind of fun, a little bit of the concept of what I mean by the selfish microbiome. Right? So, how old is the Microbiome Project? This is Ilya Mechnikov. He got a Nobel Prize in 1908. He discovered phagocytosis. Apparently he had some major fights with Pasteur. Because Pasteur would not believe that a white blood cell would eat a bacteria. He actually came up with this concept of putrefying bacteria, Clostridium firmicutes, actually caused me to get old. And really, he was the one that pushed this concept of probiotics. That's how novel probiotics, eating live bacteria. In fact, kefir, I think it's pronounced, is a fermented milk product in the Middle East. He lived to be 71 years old. Right? That was pretty cool, all right? So the Human Microbiome Project is probably conceptually goes back to that, all right? This is Theodor Eschenbach. Okay, he discovered E. coli. He had infant poop and he isolated this bacteria. The big question here is, we always hear about E. coli. Is it the most abundant bacteria in your gut? It's not. It's the one that grows on the plate. It's facultative anaerobic. It will grow on aerobic plate. Most of the bacteria in your gut are anaerobic. Right? But so this concept of actually taking, identifying bacteria and trying to look at function is important. This is Watson Crick. They discovered the structure, they defined the structure of DNA the year I was born, actually. So we're going up here in steps of finding the concept of microbial ecology to identifying the bacteria, to identifying DNA, and then moving on to essentially molecular systematics. Wally Gilbert, I worked with Wally, okay, he got a Nobel Prize with Sanger for DNA sequencing. So now that we only know the structure, we can actually do the sequencing, identify the ACTGs up there. Let's take the next step up. I worked for Carl. He actually didn't get a Nobel Prize because there is no Nobel Prize for biology. He got the Crawford Award, which is the equivalent. And I worked with him with a number. He died a few years ago. He is the father of molecular systematics. He's the one that defined the genes that we normally use to identify, to sequence and identify bacteria. Actually, all life on the planet, really. Right? The Human Microbiome Project was started in 2005. We got into microbial ecology basically in 2002, just a little bit ahead of the curve. So the idea here is we're looking at the microbial community, the microbiome, on all surfaces of the human body. Right? Now, initially, with a little bit of a essentially esoteric project, but it turns out that especially your gut flora are intimately involved in your health. Okay, and most people, okay, nutritionists have known that for hundreds of years. I just put this slide up here. So if you take a look at the gut, the concentration of the bacteria is like 10 to the 14th cells per gram. This is the highest density microbial ecosystem on the planet. You can argue that there's 10 times more prokaryotic cells than eukaryotic cells. Now they're very small, so they're not the same mass, obviously. Everything we do, okay, especially microbial calls, we're really looking at biofilms. That's really what you're looking at. So we do coral diseases. This is a biofilm right at this junction that is killing the coral. This is white plague, and this is bare skeleton. And it moves that fast that turf algae doesn't actually. This could move 2 centimeters overnight. So it's killing the coral. This is villi in your gut, and this is the biofilm that's in your gut. And we do lobster shell disease. So these are just a couple examples of some of the projects that we work on. In the lobster shell, there's a normal microbial biofilm, and somehow it gets disrupted and starts to eat away at the carcass. It looks pretty ugly. They don't taste that bad, but you can't sell it. Right? So there's this parallel between human gut ecology and all our work in the environmental systems. Right? So just to blow up the biofilm mucosa. Right? One of the big things that we have to consider here is that 80% of the human immune system is in Peyer's patches. It's a gut-associated lymphoid tissue. It's wrapped around the intestine in Peyer's patches. And I blew this up. You can actually see that the Peyer's patches are connected to the inside of the lumen. They're talking to the bacteria. Okay? And this was been shown just recently. It's not just talking to the host through the immune system, but there's direct vagal stimulation. If there are neurotransmitters that are produced by the bacteria that actually directly interact with the host. There's a bunch of things that are going on. This concept, I brought this up to try to give you the concept of dysbiosis. Normally, in the lumen, you have bacteria that essentially change depending on what you eat. Right? Then there's this biofilm, possibly a couple of layers, that these bacteria they colonize the gut and the neonate, and they're pretty stable. In fact, if they weren't stable, you'd be sick. And these bacteria now are recognized by the host itself. Right? In dysbiosis, for some reason, they're getting the lumen bacteria that are migrating into this biofilm, and now the host sees foreign antigens, and you get an inflammatory response. The classic one is inflammatory bowel disease, Crohn's disease, ulcerative colitis. Right? These are not infections. They're an immune response to foreign bacteria. Right? Okay, how do we analyze the microbiome? Right? In the past, as I inferred here with Esric, is that we culture the bacteria. Right? So in some of the high schools, they actually do culturing. You can grow them in plates with different sugars and see what they eat. You can grow them on media and see what the shapes of the colonies are, or you can grow them on selective media, and if they grow, you know, well, this is blood agar. If it grows, don't touch it, right? You don't want to put that in your mouth. Not good for you. So the problem here is that when you're doing culturing, the paradigm out there is less than 1% of the natural community that you see out in the seawater, whatever, are not cultured. Right? But we don't know what to eat. The other problem is we used to use rich media to grow E. coli, and that just kills anything in the environment. The other big thing is that bacteria are feeding each other. They're in the biofilm. Okay? you have this quorum something that's going on. A couple of examples. We had a purple sulfur bacteria that we isolated from black pan disease, a coral disease. And you look at nice, you get a nice colony on a plate, and you look at it under a microscope, there are two things there. And you can never separate them. They're feeding each other. Right? So the big paradigm shift now here is that they move to a molecular technique, because we can't grow it. We're going to use a molecular techniques to actually look at a marker, the 16 ribosomal RNA is what we use mostly, and then sequence it. So in the old days, right, back in 2000, 2002, we would amplify a segment of the 16 ribosomal RNA, and we would get different size fragments for different bacteria. We would clone it, put it to bacteria and culture it on a plate, pick these colonies and do high-throughput sequencing on a fluorescent sequence. This technology is how we sequence the human genome, fluorescent sequencing. Right? And the key here is this is 96 capillary electrophoresis. 96 samples at a time. That was a huge thing back then. Right? 96 samples at a time. Right? Now can we do it faster with less labor and lower cost? When we were at this point, if we picked 200 colonies for one sample and sequence, that was a lot of work. That was 2 runs on the floor. All right. And typically, it would cost us a couple of $1,000 to do one sample. All right. Next-gen sequencing. Now we're sequencing by computers. There's been several different generations here. I'm going to talk about the ion torrent. There's an Illumina technology, which uses a different way to amplify the fragment. Basically, in this case, we're putting on beads. And then we're going to put it on a computer chip, and we're going to sequence it on a computer chip. Right? So Rothberg is there, a very bright guy. He came up with 2 generations of this type of sequencing. The urban legend here is that he is, his father owned some kind of cement business, multi-millionaire, okay? When he turned 20, his dad gave him $20 million to go do something. So he has to get quite smart. If you look at his patents, they're fantastic, because he had a lot of money to put into the patents, right? What we're going to do now is we're going to have a community that we've amplified a marker genome. And then what we're going to do is we're going to stick just one molecule on a bead. Right? Does everybody know what PCR is? Okay, polymerase chain reaction. Okay, so we're going to stick one molecule on the bead, and we're going to put it into a polymerase chain reaction mix and put oil in it. And shake it up. What happens when you shake oil and vinegar? You get an emulsion. You get droplets of oil. You get a droplet around each of the beads. Then you run a PCR, amplify that one fragment, one molecule on each bead. So it's cloning by beads. And we have ways to clean up the beads. This is actually a real picture of it. You can see that we have oil droplets with some beads in it. Some of them have 2 beads. It shouldn't happen, but it's real. Okay? Then the concept here is we have a bead that has these adapters on it. We buy one molecule. But you end up with then getting a hairy bead. You amplify it enough that we get enough signal on the sequencer to actually see what goes in, what happens. It's sequencing by synthesis. So this is the first machine that we got in the lab, PGM. It would do, now, put things in perspective. We talked about the fluorescent sequencer doing 96 samples at a time. This produced 4 million. The newest machine we just got in the lab is 80 million. That we talked about. Now, where's the issue here? The bottleneck. No longer sequencing. It's the bioinformatics. Okay? It's the computational sciences. Right? So literally, they had a CMOS chip that had little wells in it, and you spin the bead into the well. So just to refresh everybody, okay, this is a DNA strand. When you bring in a trinucleotide phosphate, it gets incorporated, And the phosphodiester bond gets linked, and it releases two phosphates, a pyrophosphate and a proton. This is just a pH. You just measure the protons. Okay? The first generation actually measured the pyrophosphate. And again, it's just another little diagram here. So triphosphate comes in, it makes this phosphodiester bridge, the pyrophosphate come up, and releases one proton. All right? So the concept here, is we've got a plate of 4 million wells full of E's. I'm going to throw A on it. Okay? And then wherever the A is going to go in, it's going to release a proton. So if I have 2 A's in a row, it's going to give 2 protons on it. I should see twice the same. So sequencing by computer. This is just basically the schematic of the actual CMOS chip, but it's the same idea. The only reason we're amplifying is we can't pick up one proton, but we can see it change by 1,000. So we're amplifying it by 1,000 fragments on that date, so that we can see a signal. All right. So here your little enzyme, the polymerase. Okay, it's coming along, puts a base in, releases a proton. You see it uplift in the CMOS chip in that one. If there doesn't match, you don't see them. If you see 2 of them in there, you get to see twice the peak, twice the protons. Does that kind of make sense? Okay? Everybody pretty clear, pretty straightforward. I won't get into more details, but that's basically what it is. You're amplifying on a B, and you're shoving it on the plate, and you let the computer do the science. So the new machine we got is 80 million wells. It will do a human genome in an afternoon. All right? Wow. So this is just, 7 million, these are different chips on the machine. You get different read rates and different throughput. This is the old 96 capillary fluorescent machine. If you really busted your butt, you could probably get 10 million bases a year. This is doing 16 gigabases in a half. It's 1,000 years of cloning and sequencing in one afternoon. So it's flipped the paradigm of research in biology. In the past, you think of an experiment, okay? All right, let's go in and do it. You spend 9 months getting the data, and you spend a month, analyzing. Now, you plan probably a couple of months, because it's expensive, it's more expensive. So, get the data in an afternoon, then you spend the next 9 months trying to analyze it. So let's flip things upside down. This is interesting. Okay, 1977, they sequenced the first tRNA manually. They did 77 bases. It took 7 person years. When I was doing sequencing, manual sequencing, dideoxy sequencing, or fluorescent sequencing, this is actually manual radioactive sequencing. I put up about 100,000 bases in a year. I wouldn't be able to do it 2 years in a row. There's no way. When they started the Human Genome Project, they're estimating that it took 12 years, $2.7 billion, and about 50,000 person years. Right? And the original estimate was $3 billion. So that's a dollar base. They came up with a next-gen sequence, and they redid Venter's and Watson's genome in 3 months for $1 million each. Now, probably about $4,000 in a day. So this is really going towards this whole realm of personalized pharmacogenomics. Looking at your genome, seeing what drugs work better for you than another person. Okay. Foods, okay. Nutrients. Okay. What you can even get 23andMe. There's several companies out there that will actually genotype you for genes that they know are associated with different diseases. Okay. Now, I'm not sure, I haven't done it yet, I'm not sure I want to know, like 50 years, or 10 years from now, I'm going to die from a heart attack. I'd rather. So, a lot of people, all right. Systems biology, all right. This is real data, and what you're seeing here is that firmicutes they're going up and down. The bacteria are oscillating. And this poses a huge problem in doing analysis. These are nonlinear, non-parametric systems that you're trying to model. And this is a huge, huge problem. In this case, what you're looking at here are short-chain fatty acids that are, these are genes that are associated with circadian clock. And they're looking at them with or without short-chain fatty acids. Right? And this paper was interesting because if you take a germ-free mouse, right, not a biotic mouse, and you give them either a low-fat diet or a high-fat Western diet, you're going to see that disruption of the circadian clock, that the mice don't get fat. And germ-free mice, they put the mother in a germ-free environment. Okay, they deliver the pups by cesarean section and put them in the germ-free, so they don't have any bacteria. They don't do that well, frankly. Okay? But if you take conventionally raised mice with the gut flora, all right, you disrupt the normal diet. Okay, they look normal. On high-fat Western diet, high fructose corn syrup, and high cholesterol. You disrupt the circadian clock, and the mice get fat. So they need the two of them together. Okay? So there's dozens of papers out there associating the gut flora with obesity. Right? And this comes in with the whole idea of nutrients, modification of your diet, probiotics, okay, fiber, okay, break the cycle. All right? So the point there is that it's the modeling that's the big issue. The sequencing has become a commodity. Right? You don't even have to have a machine with that. You just send it out. They send their services all over the place. Right? You get the data back. What do you do? Okay, so it's the mathematical modeling that's the big thing. All right. This is just a quick little blurb here. These are the also about pigs. These are the be careful how I say it. Okay, I was down in Florida and I said the Spanish pigs. They got upset. These are the original pigs from Spain. They're wild. If you go to the islands like several different islands off of Georgia, they run free. They are what we call a hardy phenotype. They utilize food well because they're at the mom. Right? If you take them into the barn and feed them ad libitum, they die. They get so fat they can't walk. Heart disease, metabolic syndrome, okay, a bunch of things. Okay, diabetes. And the same thing applies if you think there's a parallel here with Native Americans, Hawaiians. When they were a hardy phenotype. They were living off the land. When you bring in a Western diet, okay, they all get this. All right? It's a parallel. All right. In this case, it's interesting. They fed them a low-fat diet, a high-fat diet with a probiotic, and just a high-fat diet. And they were hoping that the probiotic would actually help maintain the weight. Right? Sorry. They measured, their joke is they used everything but the squeal. They took all different tissues, they did real-time PCR assaying different levels of messenger RNA, they did metabolome, they did a whole immune system, markers, everything. This is a PCO plot. Just think of it as these coordinates measure the variance of the data. So if the variance of the data is the same, they're clustered together. So clustering, what we see is all the controls are clustered here. All the high-fat diet, with or without the probiotic, are over here. So the high-fat diet overwhelmed any effect of the probiotic. And this is a big, big problem. I was on a couple of class action lawsuits for probiotic cases, and this is the problem, that they're not going in scientifically testing out the effect of probiotics. They're just making all these claims. Right? I have to be careful what I say here. Dan Active is one of them that lost. Okay? This is the interesting one. Dan Active lost a class action lawsuit. They lost $60 million. Went on for 5 years. It had to cost them several $1 million in lawyers' fees every year. Okay? They didn't care. When they released Dan Active, it was the biggest product release ever in the United States. They were selling $2 billion worth of product. Let it pass. They didn't care. So they eventually did change their advertisements. They cannot say clinically proven. it may help. All right. I talked about modeling. So in this case, what we're doing is we're looking at the correlations between bacteria and gene expression and immune markers. So think of it as this: a node is a feature, right? An edge means there was a strong correlation between the two. So these are what we call correlation networks. And yes, okay. Here, the orange are bacteria, and these are all your liver enzymes are correlated. Well, they better be. You got cirrhosis, all right? Your enzymes in your liver should all be correlated. Adipose tissue. This central node here is actually the brain. That gene right there is leptin, which is the main key for satiation. All right, which just makes sense. So you actually start to look at essentially homeostasis here, all the interaction, and the bacteria stuck in the middle. The bacteria are talking to all these systems. And so you can think of this as, this is a link between the brain, okay, and your gut. We're talking, this is modeling the interactions between. Alcoholic liver disease. Okay, so this is a big project we've been working on for a number of years now. Normal liver, fatty liver, all right. This is basically steatohepatitis. It's NASH, non-alcoholic steatohepatitis. Okay, we're really getting damage, and this is cirrhosis. I was at a meeting, a NASH meeting in France, in Paris. The Frenchman had a real funny sense of humor. So we went to a real fancy restaurant with all the senior people. Actually, Arcade National, to, you know, it's all gold rooms like Napoleon, whatever, right? Stay fancy dinner. The appetizer was foie gras. Okay. And most of the Americans didn't know that they were eating. Okay, fatty liver. Anyways, this is a big epidemic. All right. People don't know they have it. Somewhat associated with obesity. But it's even more than that. In India, there's a phenomenon called lean NASH. People that are thin, look thin, they have it. So you have to be careful because it will progress. And part of the problem right now is to really diagnose this, you have to go in for a liver biopsy. It's just not anyway. I'm not interested. So these are bacteria community, the average bacteria for the controls. And the key here is the yellow, the Bacteroides. Okay, this is the lactose. These are the main bacteria. The Bacteroides are going to be the major dominant species in your gut. All I want you to see is as we go through this, as you progress through the disease, the Bacteroides are getting higher and higher. All the other ones are getting less. So there's this biotic shift. So we can do the correlation networks here. And again, now we're looking at the correlation between the gut-liver brain axis now. Okay, because the liver's involved, intimately involved. The liver basically detoxifies everything coming in through. So if your bacteria are producing toxins, okay, that's where it gets taken care of. If you got cirrhosis, you have major problems. I have this here. Okay, so take this one step further. This project is hepatic encephalopathy. People that have cirrhosis can't metabolize those toxins from the gut, and they can't drive a car. Okay, they're that impaired. In fact, one of the tests is they sit them down in a simulator and they count how many times he drives off the road and see. Right? So one of the main arguments is that the bacteria producing ammonia, it goes directly across the blood-brain barrier to your brain. Right? There's a lot more that's going on. This study was interesting in that we took people that had hepatic encephalopathy. This same group gave them rifaximin, which is a non-absorbable antibiotic. So whatever the effect is, it's with the gut. Right? This is the bacteria before the rifaximin. Again, the Bacteroides is the major one. This is after. I ain't seen much of a difference. The bacteria really didn't change. However, if we do the correlation networks of all the metabolites, the green are serum, yellow are urine. Right? No, the other way around. Serum is yellow, green is urine, all right? This is before, and that's very complex, okay? This is after it changed. And this is the difference. Statistically, we can go through and see what statistically changed between the two states. So this is the correlation network that has changed. So now we can go in, drill in here, and see what compounds, what bacteria, okay, are being changed. All right? This is what's the same, so this is probably the network for cirrhosis. All right, I'm going to say this again. So now we can take a look at this and look at the interaction between the gut, the brain, okay, looking at the microbiome at the top. However, correlations are not causality. Here's the boss yelling at Dilbert. Says every time I leave the cubicle, I get a nasty email. Okay, Dilbert's in the back, Wally's in the back pushing the send button. Okay, Wally's saying no. Correlations don't imply causality, right? But it is this idea of knowledge discovery. This is a hypothesis that now we can go back and change. This is a cycle of discovery science versus hypothesis driven. It goes around. All right, selfish microbiome. Here's some fun. This is a house microbiome. You want to swab all the surface in the house? See, I knew we should have did this after. Okay. The microbiome is the same in the whole house, and it is similar to the people in the house. And it's the dog too. Well, of course the dog. You got a dog. If you have any dog, you know what that's going on. All right. So the house microbiome is the same. This is your hand, foot, nose. They're all the same as well in that house. This leads into before dinner. I don't know. This leads into the issue of fecal microbiome transplants. All right. Okay. It's actually an FDA certified treatment now for people that has Listeria difficile. They give the microbiome transplant from a member in the house, a spouse or sibling. It works. Okay, cures. All right. So you have this dysbiosis, and now they do it just with an enema. There's actually now recupulate pills. or Frapsol. There's a company that's selling capsules, okay? All right. So it's becoming maybe a better way than probiotics. All right. For probiotics, okay, you're not sure that it will colonize that person's gut. But if it's from something that's similar, a person similar to that. Anyways. The microbiome cloud. Put people in aerosol chambers, filtered the air, looked at the bacteria. People are shedding bacteria all the time. Now the problem is inflammatory bowel disease contagious? Can you pick it up, that dysbiotic community, from someone else? I don't know. Anyways, the interesting thing here, remember Charlie Brown and Schultz? Okay? Pigman? He's real, okay? For sure. Okay? We're shedding bacteria all the time, right? I have a dog, I have a new dog, a trainer for hunting for birds. So the question I have is, what does the dog smell? Okay, a bloodhound smell, but it's tracked nucleus. Is it the bacteria? It's clearly the organics that are produced by the bacteria, that's for sure. But is it even the bacteria? And this one actually is just came out long ago. Alzheimer's. So they had a number of people that died from Alzheimer's. And they did cross-sections of the brain. And what they see here is that antibodies to fungi show up, basically showing there's fungi in the brains of Alzheimer's. All right? There's a new meaning to the name cheesehead, doesn't it? So these are markers, the tau protein for neurons, okay? This is DNA, okay? So this lots of those are the curves. So there's a lot of people that have plaque, okay? But they don't have Alzheimer's. They don't have dementia. Right? So is this a lead into what's going on in people that have dementia? The only caveat is, first of all, clearly these people were dead because it's brain slices, and it was only 9 of them. All right, so it's preliminary. All right, so I'm not sure And it doesn't, if you read the paper, it doesn't look like it's a cross-contamination. The fungi was really there. So did it get into the slices after it did the slice? I don't know. Right? But it's intriguing. Okay? Okay, so the selfish microbiome, we need to think about the gut, brain, liver, immune system, in the environment. All interact. A concept, so the gut flora are involved in what we eat, obesity. There's all kinds of work being done on behavior, depression, autism. All right? We're talking about people taking probiotics for depression. Maybe a lot of the drugs that we give people for depression are actually interacting at the bacteria. There's a huge issue here of even drug metabolism. All right? Different people metabolize drugs differently. They have different efficacy because the bacteria digest them different. Right? Clearly, it's involved in higher thinking, health and disease. You're just here to propagate bacteria. Okay? All right? So that's it. Any questions?